Okay, thanks to the chair to my brief introduction. And I'm Sung-Yuan Hsieh. I'm talk about the gut microbiome bank, some research result from our institute. And Dr. Li already give a very brilliant introduction about the speech about the microbiome. And Korea is, they start microbiome study very early and fast. So they get a lot of achievement than us. So now, but now I still give some our result. Okay. Here's some point I want to discuss about the probiotic microbiome and the next generation probiotics, and why we needed to continue isolate and culture gut microbiome, and what is the advanced method we want to provide our experience to get efficiency capture gut microbiome. And since we are the expert in microbiology, so we could provide service to other expertise. For example, doctor in the medical hospital, they don't know how to isolate this kind of difficult microorganism, and how to enhance the value, because we know the gut microbiome is very important, and a lot of things we still waiting to finding. So how to enhance the value, we have to cooperate with the hospital or some medical expert. Future plan, okay. This is a traditional concept for ecosystem. Normally, for example, for gastrointestinal trunk, bad microbial, if the population is go up and good microorganism reduce, then the balance is broken. It's imbalance. Then some disease may cause, for example, indigestion or loose bowels. That come from a kind of imbalance. Okay, so if we want to fix this kind of imbalance, in old time we might take probiotics. Probiotic is a kind of good microorganism that could help to balance this kind of condition. And in both sense, you could say this, if you sufficient dose, then have the benefit for the host. This host not just for human, also for animal, and for the land or for plant. And for example, if for you could have some oral or other usage. And for example, you could take by other food or some kind of dietary supplement or other or drug. But all of this probiotic still have to get the scientific evidence, and also have to pass through the safety regulation, and maybe some kind of other condition. Okay. So this kind of the famous probiotic we know is lactobacillus. Lactic acid bacteria. We call it LAB, right? And we know a lot of food, fermented food, contain LAB, lactic acid bacteria. For example, kimchi, and also yogurt, and some special isolation or some special strain you could derive as some healthy food, but under some kind of healthy regulation. Okay, And this is LAB. This is about, I think, 20 years ago, the research is an international project. A lot of countries involved, China, Japan, Taiwan, Indonesia, and Thailand. And they, some microbiologists, they investigate the flora of the, from the human fecal and other source. And the result is quite interesting. And some fungus, for example, bacterial and phrages, this one is common, happen in all the country. And this one is bifidobacteria is the same. But some, for example, this one is porvotelasis. This one, some country is very, the density is low in Japan, but some country is high. This one may be concern the diet, because each country, they have the different diet. Okay. And till now, we know our technology is improved and our knowledge is improved. So we know better than before. For example, we know those fungus is harmful, or Streptococcus and bovis. And for example, enterotoxic producing strain, and also Corybactin producing strain of E. coli. And those may be harmful. And so this one, and those is, we think, is healthy strain, include AKK, Acmenia musciphylla. And we also know better if this kind of imbalance that induce quite a few disease, not just the concern with our digestive tract, also in other people. That's because some kind of intestinal permeability imbalance, or some kind of vitamin or short-chain fatty acid is reduced, and also inflammation from the induced by those bacteria. Okay. Since we know some kind of fungi, some kind of bacteria or microorganism is very is concerned or useful to some disease, so the people try to use those beneficial bacteria to become another kind of probiotics, because they don't have no history to use. So not just like this bifidobacteria, lactobacillus, they have long history to use. So they are quite safe. And those we don't know. We know they may be useful or functional, but we don't know the safety. Okay, so we call it, probably could be a next generation probiotic, or a kind of live biotherapeutical drug. Okay, based on the gene sequencing and analysis improved from 2008, there are few large projects to sequencing the microbiota from the human. For example, from the intestinal gut or skin or mouth, everywhere, and following other, this one is from America and other from Europe. They find the microflora is connected, correlation with some disease. For example, some heart disease and some other liver disease. However, that is correlation. We don't know which one induced, which is the result, or it's the cause. For example, we find the flora is different, but we don't know this flora is the result because he is fed, then get this kind of flora. And this one is thin, so get different kind of flora. We don't know this one is the result or is caused. So if we transplant those flora into a experiment animal, for example, rat, then get fed rat, and this one get thin rat. So we know the flora is the reason to become the fed. However, this one is a flora. Which strain or which species cause, we don't know. So now, at least now we know this is causation. Causation induces the different disease. And next, we want to know which fungus may cause this disease and which maybe metabolite cause this disease. And here is some putative mechanism was proposed. Probiotic may, with the pathogen, maybe they produce antibiotic peptide to competition or reduce the number or reduce the pH in our stomach or in our intestine. And competition, there are several competition or inhibition. This is the first colonization resistant. The second is the immunomodulation. Some strain already know they could reduce some factor and received by the dendritic cell and maybe influence some immunocell, then do some immunoregulation. And the third, they maybe produce some metabolite. Sorry, here. Enhance gut. Maybe I could enhance the intestinal barrier, because if intestinal barrier is unbalanced, some fungus or some things will become loose and could penetrate. But probiotic, if we don't know its safety, sometimes it causes some risk or dangerous. For example, they maybe could transfer, pass through the epithelial barrier, then become the system infection. And or another risk is this one, maybe transfer some resistant gene. This resistant gene could resist about the antibiotics, then to the pathogen. Then pathogen could become the resistant for the antibiotic. And some maybe could produce could cause the tumor. Okay. And another mechanism, some gut microbiota could produce TMA. TMA is the trimethylamide. Then if this one, the concentration is high, then could pass through the liver, and this liver will transfer to TMAO. TMAO will induce some blood vessel disease. So this is a metabolite to induce some heart disease. And till now, a lot of clinical trial has been doing at this moment, especially for this Clostridium difficile infection. a lot of clinical trial already doing by this kind of, we call it fecal microbiome transplantation. You use this kind of cure method in clinical trial. And even FDA approved some drug is for the FMT use. Okay, and if this one is already successful in many cases, could recover some instantial disease. However, FMT donor have to choose carefully. For example, donor have to recruit and selection his or her healthy condition, and then choose suitable donor. Suitable donor then could donate their fecal, and normally fecal could through some treatment then become some treatment dosage could use FMT treatment. But in the future, maybe they could isolate from this fecal, isolate the fungal, or choose some, or even propagate, and then just keep for cure some disease. And here is the, at this moment, people is first generation LBP, we call it live bacterial product, biotherapeutic product. And FMT is first, at the first stage, but second generation could be come from the known species, for example, AKK. And the third maybe come from some unknown species from gut microbiota. And this one is a French researcher, and he is a pioneer in cultureomics for microbiota, gut microbiota. And he thinks culture is most important, because if you get a culture, you just could check the mechanism, and also to become some kind of treatment, for example, some kind of medicine or diet or healthy product. For example, AKK, now we know this one is type strain, and different strain may have the different function, or strong or weak function. And till now, people think still have a lot of strain uncovered by the present technology. And we only found about 40% strain, so still uncover a lot of strain waiting for find. And here is the important why, important reason why we use the cultureomics. Because if we use the DNA sequence, DNA method, if the concentration, if the population is too slow, lower than 6 times, you cannot detect by molecular method. But if you lower than 10 times, then you still could have chance to find. So this area is a lot of species you can find, you cannot detect by molecular method. So Professor Rout, he developed cultureomics for microbiota. He used more than 200 conditions to find gut microbiota. And he got 1,500 strand. But this one is incredible. So other labs that cannot use this method. And next, they refine the method. So it's now they develop about 16. It's affordable. 16 best cultural condition for culturomics is affordable. So, and except the culturomics, you use the affordable medium method. Another important tool is the multi-TOF. Because multi-TOF, it could very fast and high throughput to identify the strain. Because if you get the colony, we don't know set strain. If you get a colony from a fecal sample, maybe you have 2,000, 3,000, 5,000 colony. How can you identify? It's no chance to identify 5,000 from one sample, right? Dr. Lee said he get 800 people sample. So you have to find a fast identification method. And multi-TOF is very useful, and you could get you get a result about half hour, then you could get about 100 strand, the result. It's very useful to replicate. Otherwise, if you repeat to identify same species or same strain, it's useless. And if you use the multi-TOF, if you get the same pattern, then you discard. You don't need to next, you don't need to identify so many strands. Okay, so this one is quite useful. Another useful is this single cell dispenser. Normally, if we use the plate, if you dilute sample, then take some volume to plate, your agar plate. You get a lot of colony, right? Maybe 1,000. But this 1,000, you cannot make sure every colony is pure. Sometimes it's several different species aggregate together. Then you have to purify. You have to purify several times. Then you make sure it's clear. It's one species. But this one, you could dispense each cell in one plate, in one position of a plate. And the plate is like this. Oh, sorry. This is the plate. And each plate you could have many. Each dot is come from the one cell and become one colony. Then this one is 384, and also very fast. About if you get one sample, normally you just dilute to certain concentration, and that automatic to put cell in this plate. Normally we use 10 kind of media. So you just identify this 3,000 colony. Normally half could grow up, and some cannot grow. So about 1,500 colony. But this one, 10 minutes. So it's very fast. And you don't need to do replication. So it's fast. Okay, I have to go fast.
Yeah, you have 3 minutes left.
Okay. So this is the result. Oh, sorry. This is the result we get. 1,000 strand and 200 species, and we believe 50 is novel. And this one is we get a sample from different, for example, human and human and pig, and human and mouse, and different animal. Then some strain is come from human, also come from the pig, something like that. And a few years ago, our government supported a project. We call it Microbiome Core Facility Project. And BCRC is in charge of the culturomics. And some is FMT, some is animal model. And we are in charge of this one. And this one is some result. We is a kind of customer service, and we get some result for customer. And this one is we cooperated with some medical institute, and we found 120 species, and at least 10 species is concerned with the limb association bacteria, and do another experiment, then get a good result, confirm the function. And this one is experiment concerned with we want to find some strain concerned with fast piglet could grows fast and fat. And so we get 70 species, and one species could become treat as a diet and get the good result. This one is 11, and this one is treatment is 18. So it's piggy could go fit and go fast. And this one is another experiment. We induce the mouse get the qualities. Then this strain could protect the mouse against his qualities. And this one is the current status. A lot, more than 100 company, they start to study microbiome therapy, and some has become the product. Especially this one is a Korea company. They also already get the FDA approved. It's called new dietary ingredient notification. And here, there's some challenge about microbiota therapeutic identification and safety and regulation, and identify association with disease status. And so here is still a lot of challenge. And here is we cooperate with the Changgen Hospital. They are very good in FMT treatment. And this cooperator is, we are looking for super donor, the fecal from super donor, and we want to isolate the strain from this kind of sample. And at this moment, this is a 4-year project, and we intend to get 1,000 strain from a super donor. And at this moment, we get this is our result, present result. And this one is we cooperate with Taiwan, National Taiwan University Medical College. We are looking for some strain could concern with the allergy. Yes, and this is the strain is with the anti-allergy effect. And this one is we found is a new species. And this one is AKK concerned with some anti-hepatitis B virus effect. And this one is also published. And Okay, this one is a species, a strain is anti-cholestis effect. And we found in some case, BCRC, this one we at this moment still call it AKK, but different species. But this one is the type strain. They have the different effect. So I said for this kind of live bacterial product depends on the strain. So this one is our plan. We want to find within the 5 years. We want to have this kind of product in the market, and within the 10 years, we think because the safety issue is have to recover. And we have the different core technology and is willing to cooperate with some industry or some culture center as well. Okay, yes, my presentation. Thanks.